We first met at University of Alberta in Canada when we were both doing our PhDs. After completing her PhD at University of Alberta, Melissa performed a postdoc at Harvard Medical School before returning to Canada to take up a faculty position at Carleton University, where she is now. She's now a tenured faculty member and associate professor in neuroscience. So Melissa's research has been primarily focused on hypothalamic system to regulate body weight, with a particular emphasis on understanding the neural circuits controlling food intake. She is very interested in knowing how physiologically experience, such as anger, fear, anxiety, and also specific nutrients like fats and sugars, can alter these circuits. One of the peptide systems she's currently working is melanin concentrating hormone, and she will be sharing some of her findings related to these in their presentation. She has a strong foundation in cellular neuroscience, built through training in electrophysiological methods that she started already in her PhD, and this will be evident in today's talk, where she will present an adaptation in one of these techniques. Additionally, her lab uses a variety of tools, such as virus for track tracing and neuroanatomical mapping, and also optogenetic and chemogenetic tools to manipulate neurocircuits and explore their contribution to behavior. So really, Melissa, it's really a pleasure to have you here. So as we just said, our first external outside of Europe to be here. So I'm really, really happy. The floor is yours.
Hi, everybody. Thank you so much, Sarah. Thank you very, very much for having me. It's a real honor, and especially to learn that I'm the first person from across the ocean to be here. And I'm really honored to have the opportunity to contribute to this lineup of presentations on current research techniques. And importantly, to all the students, or even if you're not a student and just interested in learning about current research that's happening today, hello, and thank you very much for tuning in. I'm learning that this is evening time, so good for you for being here. The research technique that I will be speaking about is an adaptation of patch clamp electrophysiology in fresh brain slices. And I will make the case for how it can be used in identifying drug targets in the process of drug discovery and development, because I was told that this is sort of the focus of the Braining Club. So just to kick things off, I want to express that electrophysiology is a technique that allows us to determine if a cell is functionally active. And that is helpful in drug discovery because it tells us whether the cell will be inhibited or excited by a compound, and in turn, whether a drug will depress or enhance neural activity. Now, we can perform electrophysiology on cultured cells or from primary neurons that are isolated from the brain. But what I will focus on today is the use of electrophysiology in the intact brain slice. So before I launch into the presentation, I just want to give you a preview of what this technique is. So this particular technique, I think, has distinct advantages from cultured systems because when you're in a brain slice, you can retain all of the endogenous neural circuits and targets while isolating the contribution of just that individual cell. So within the brain slice, we would perform whole cell patch clamp recordings from a single neuron, and electrical activity from a cell looks something like these. It looks like little spikes up and down from the membrane. And traditionally, one would bath apply a drug to the entire slice, so that all of the cells within the slice would be responsive to the drug. But the problem or the caveat of that is after applying the drug just once, you can't use that slice anymore, because the other cells, even though you didn't record from it, had already seen the drug. So you're going to have some kind of a confound. So the adaptation that we've made will be to puff a drug by moving a puff pipette next to the cell that I'm recording from, and then delivering a small puff of the drug to that cell, and then seeing if that puff of drug will change the spike firing or the membrane potential of that neuron. So the drug will puff, and then we could record the change in electrical activity from that cell. Okay, so the benefit then is because your drug is applied really only to a small area of the slice, when you're finished with this cell recording, you can remove your patch pipette, bring in a new one, move to a different cell in a slightly different space in the slice, and then just move your puff pipette close to your new cell, right? And then you can repeat that process again. So one more time, this is you move the puff pipette next to the cell that you now want to record from. You apply your drug recording, you apply the puff, you see a change in the membrane potential, and then as the drug gets washed off very quickly, the cell can come back to its baseline. And this technique that I'm going to talk about, I've just called it patch and puff. So now, before I launch into more details about this approach, I wanted to take a step back and point out where this technique might fit into drug discovery. Okay. I'm going to talk about the drug discovery process, so the stages of drug discovery. And this particular presentation is going to focus on discovering targets. I will then show you an example of how I have used patch and puff application in my own research, and that is to identify target sites of melanin concentrating hormone action. I call it MCH. And then we will discuss the merits of doing brain slice recordings, and then close with a stepwise protocol that you can take with you to adapt to your own experiments. So first, the different stages of drug discovery and development. There are really 4 or 5 stages. The first is target discovery. Target discovery is where most academic researchers like myself fall, and this is where drug discovery starts. It starts with a systematic, stepwise investigation to find a biological target that is involved in disease. So once a potential target is identified, then we can move to preclinical testing. And this involves either in vitro or in vivo testing. In vitro can be on cultured cells, but in vivo testing is typically completed using animal models. Rodents are likely the most common. And should that drug be passes through the preclinical research stage, then it can move on to clinical trials, which then involves testing in humans. And within the clinical trial process, each trial is actually divided into several phases where the number of people that's included in each phase will continue to increase. And that's really important because testing a larger and larger sample as we go along allows us to get a better estimation of the side effects that the drug might have, especially when it gets rolled out into the public. So after clinical trials, then there are actually 2 additional steps, which I've just lumped into one because I won't be focusing on this, and that is obtain regulatory approval for the public to use that drug. So in Europe, you might be familiar with the European Medicine Agency or the European Commission, and in the United States, that's the FDA or the Federal Drug Agency, and in Canada, where I live, we have Health Canada. And then once the drug goes to market, it continues to be monitored. And I think that's actually really important because as a drug gets marketed to even a larger population in the public, then you open ourselves to the possibility that even more side effects could emerge. And should those side effects emerge, then these regulatory agencies have the opportunity to consider or reconsider whether this drug should remain in the market. And I think that this is really essential because it will ensure that as patients take these drugs for a long term or chronic use, we want to know that the drug is safe even for long-term use, right? So that's just an overview of the stages of drug discovery, but my own research pertains to just target discovery and preclinical research. And this particular presentation is going to focus on a technique that we can use in target discovery. So a little bit about myself and my research. My research for target discovery is to study the neurobiology of obesity. The root cause of obesity is actually in the brain. Body weight regulation and maintaining a stable body weight is very difficult. And maintaining a stable body weight is really achieving a balance between energy output and energy intake. Okay, so energy intake would be the food that we eat, but energy output is how much energy or calories can we burn. So total energy expenditure actually is comprised of 3 to 4 components. 60% of our energy output actually comes from our basal metabolic rate, which we can't really change, unfortunately. And 30% of our energy expenditure comes from exercise and non-exercise thermogenesis. So exercise would be you're going for a walk or you're going for a run, you're doing a physical activity. Non-exercise thermogenesis is, for example, if I'm sitting in my chair and I'm feeling very fidgety, I'm burning very little energy, but still it's a form of uh, energy expenditure. And then 10% of our energy expenditure comes from the thermic effect of food, which is the energy that we burn by digesting the food that we eat. Okay. On the other side of the equation is food intake, how much calories we take in. And there is only one way to take in calories, and that is to eat. So on each side of this equation, there are really only two components that we have control over. Okay, on the energy side or energy output side, it's exercise. And on this other side is eating, right? So the ability to control our body weight by exercising is unfortunately and sadly not really due to exercise because this is only half of the 30%. But this other side of the equation, our food intake, contributes heavily to our ability to regulate our body weight. And actually, the control of appetite and food behavior is one of the most lucrative markets for pharmaceutical research. You have probably heard of recent drugs like Ozempic, because a lot of celebrities have been taking this to lose weight, and it is indeed very effective. But in fact, the three drugs that are currently approved for treating weight loss or for providing weight loss is Wegovy, Zepbound, and Saxenda. Um, and these drugs are all based on the GLP-1 receptor agonist. So GLP-1 stands for the glucagon-like peptide. Um, and the interesting thing about these three weight loss drugs is, yes, it is extremely successful, but unfortunately, despite its success, still not all patients will respond, right? And because not every single patient will respond to this drug, there is still a need to continue R&D to treat these metabolic diseases. So just as an example, Wegovy, for example, a research from 2022 in JAMA, they showed that at patient follow-up at either 3 months or 6 months, um, not every patient, right? So this is the percent of patients that are responding to this weight loss medication. And in general, for metabolic disease, it's thought that achieving 10% of weight loss is a crucial marker for really gaining the benefits for your metabolic health. And at 10%, you can see that even at 6 months of follow-up, only about 60% of patients are responding. And so there is definitely really high interest in continuing R&D for obesity and metabolic disease. And some of the recent efforts actually have been to find compounds that will act as dual agonists. So Wegovy acts on GLP-1 receptors, and one very popular drug right now is tirzepatide. And tirzepatide, or its pharma name Zepbound, the drug name, it activates GLP-1, the glucagon-like peptide receptor, and also GIP. The long name of GIP is glucose-dependent insulinotropic polypeptide. It's a mouthful, but we just call it GIP for short. So this is a dual agonist, so act at two receptors, and together they show that to get that 10% weight loss, about, oops, I feel like everything has frozen on my screen. Or just my mouse. Okay, here we go. Sorry. To get that 10% of weight loss, now we increase from 60% to about 80% of patients are responding. But then you can see there's still 20% of patients that are not responding, unfortunately, to these drugs. And for that reason, the R&D is there to actually improve and continue to refine drugs so that we can hopefully have solutions for everybody. My research program does not focus on GLP-1 agonists, but on a peptide that's produced in the hypothalamus that I'd like to make the argument could be a pharmaceutical target. Okay, and that peptide is called melanin concentrating hormone, or MCH for short. So melanin concentrating hormone MCH is made only in the hypothalamus. So it has a really unique distribution in the lateral hypothalamus. And I'm particularly interested in MCH because they have quite widespread actions. So, for example, it acts on really fundamental behaviors and has fundamental functions to regulate energy balance and also sleep-wake behavior. It also impacts motivated behavior. So, on feeding, on drinking, and also mating behaviors. And MCH can also impact integrated behaviors for predation, on mood regulation, on olfaction, decision making, and also maternal and sexual behavior. I'm particularly interested in MCH because of its role in controlling food or food intake. MCH, when delivered into the brain, will stimulate feeding and promote reward feeding. And the loss of MCH, so when you don't have MCH or when the neuron is ablated, the mouse becomes very lean and hyperactive, right? So antagonizing the MCH system could produce leanness and, in a way, be a target for treating metabolic disease. So the MCH system is an active pharmaceutical target, firstly, because antagonizing the MCH receptor has potentials as an anti-obesity agent. But I'd like to point out that I think what is unique about this system is its potential to also address mood disorders like anxiety and depression. Metabolism and mood disorders are inextricably linked, and several antipsychotic medications that treat mood disorders can also lead to, unfortunately, significant metabolic challenges. So, the possibility for MCH antagonism to address both metabolic and mood issues is, I think, very attractive. So, for that reason, this is why I've expanded my research program to really focus on MCH. So MCH is made in only one brain region, but it has really diverse and widespread projections. And the reason that it's widespread is because it's probably targeting several brain regions. So I did an experiment where I delivered a virus encoding synaptophysin to an MCH-Cree transgenic mouse so that this virus will be taken up only by the MCH neurons. And synaptophysin, you might know, is a protein that tends to be on the axons and nerve terminals of the axons. And so when that cell, the MCH cell, takes up this virus, it's going to be delivered to the far end of its axon. So in that way, we know where it's going to end up. Where are the projections of MCH neurons going to terminate? Who are they talking to? And in this image, I'm showing that we see really dense MCH fiber projections in the forebrain of the mouse brain, but not so much in the midbrain or the back of the brain. So the fibers from the MCH neurons come up towards the front, up along the medial septum, and then branches out to the lateral septum. So if I just have a larger picture, this is the midline of the brain. The fibers are coming up along the medial septum, and then I can see that the end terminuses are in the lateral septum. So I should actually mention that until my observation that the main projections from MCH neurons are in the lateral septum, a lot of the field had been focusing on the nucleus accumbens as a target region for MCH action. But we never saw a strong projection or even at all in the accumbens. And so I was really quite excited to discover that MCH projections are going to the lateral septum because we hadn't seen this in our field before. So if we zoom in on the lateral septum even more, what we can see is Bhutan-like structures, and these are the black dots that are enveloping these purple cells. We can see that there's a cell, and then there's black dots that are covering the cell. And in this paper, we showed that these Bhutans are actually directly releasing neurotransmitters onto that cell, and it forms a strong monosynaptic connection through glutamate release. So these are peptidergic cells from the hypothalamus that can also release glutamate, and that glutamate innervation is what directly connects the MCH neuron to the lateral septal cell. But what I was interested in is determining if that lateral septum cell does it also respond to MCH, the peptide? So In my lab, Michaela Payan, she's my PhD student, she completed this set of work where we investigated if the lateral septum has MCH fibers within in that region. So here we are doing a stain, an immunostain for MCH in the lateral septum. And we've done analyses in the dorsal part, so the top part of the lateral septum, and compared it to the bottom or the ventral part. And in the dorsal part, we actually didn't see a lot of MCH containing fibers, so very few pink colors on this page. But in the ventral parts of the lateral septum, we saw much stronger fiber labeling that contain MCH. We also do anatomical mapping work where we can take these fibers and then transfer them onto atlas plates so that we can generate a resource that anyone interested in MCH or anyone who's interested in the lateral septum could use to guide their own research. Okay, so the lateral septum has fibers that has MCH, so maybe MCH is released there. But what's really important for having action is that it needs a receptor. So the receptor for MCH in the rodent brain is MCH receptor 1 or MCHR1. So we looked first to see if there's MCH receptor gene expression. So we performed RNA-scope in situ hybridization to look for the mRNA for MCH receptor, and that's what's labeled in these red dots. So the open arrows are pointing to cells that have low quantities of MCH receptor mRNA. So you can see there's only 2 dots, for example, here, but then a white arrow is pointing to a cell that has a lot of copies of MCH receptor mRNA. And we noted that in the ventral regions of the lateral septum, there were more cells that had more copies of MCH receptor gene expression. And we just plotted the cells that have the high, so the white arrow cells, onto our atlas plate. Okay, so great. There's 5 MCH fibers, there's gene for the MCH receptor, but does it actually make the protein? So we were lucky to find a MCH receptor antibody. And one interesting thing about the MCH receptor is that it's expressed on the cilia of a neuron. So I'm not sure if you knew this, but every neuron has a cilia, like a little antenna that sticks up from the neuron. And it's not really clear what the cilia is doing, but I think it's actually really important for detecting peptides or other nutrients that's in the extracellular matrix. So again, in the dorsal region, so the top part of the lateral septum, we didn't see very strong expression of MCH receptor, but the MCH receptor protein expression was quite a bit more dense in the ventral regions. And again, here's the map of that receptor distribution. So we had also counted the proportion of cells in the ventral regions that expressed MCH receptor. And this is just only 17%, right? So if I wanted to see if a cell within the lateral septum is functionally responding to MCH, I have a 17% chance of landing on a cell that expresses the receptor. And I'm trying to foreshadow why this is actually really important for the technique that I'm going to introduce to you in a few minutes. But first, I wanted to just show the argument that the MCH fiber distribution and MCH receptor distribution within the lateral septum is really quite unique. It's not the same throughout. So these are maps of the lateral septum from rostral to caudal. And you can see for both the fibers and the receptor expression, the distribution is different. The distribution peaks typically towards the middle of the lateral septum and is fairly low in the rostral parts or the caudal parts. And even within the lateral septum, the distribution is very unique. MCH receptor, for example, hugs the lateral aspect of the lateral septum, and the medial portion does not have as much receptor, if at all. So to convince you that there's this good overlap between the fibers and the MCH receptor expressing cells, I've just overlaid the fiber maps and the receptor maps onto each other. And in doing this overlay, what I've shown is potentially some hotspots for where I should be doing my recordings to find MCH action, right? So I think this bottom target, the ventrolateral portion of the lateral septum, has fibers and also receptors that could detect any MCH release. So we then do patch clamp recordings from this hotspot in the ventrolateral region. And this picture is just an example of what I might be seeing under the microscope. So this is a picture of the lateral septal brain slice, and then at higher and this is my patch pipette approaching the ventrolateral region. And then this is a higher magnification image where you can see individual cells. Here is the cell that I'm recording from. And sometimes after our recording, we do post-hoc staining, so we can fill our pipette with the biocytin to just label the cell that we actually recorded from. And this is very good so that we can confirm that we in fact recorded, or at least it's good to confirm where you recorded from. Okay. So what did MCH do to the lateral septal cell? So here is an example trace of the cell recording, this patch clamp recording. This dark upward spike are action potentials rapidly firing. And the red indicates the time that I applied the MCH drug. So we can see that when MCH drug application comes on, it can actually eventually silence the spike firing of that neuron, and that silencing is quite long lasting. This particular recording took about 60 minutes, so almost an hour, for the drug to fully wash out. And if we are patient enough, and if the cell lives long enough, we will eventually see the spike firing return. So I've just quantified this observation here in this time course. So here's our stable recording. At the beginning, we applied the drug and MCH hyperpolarizes, so it depresses the membrane potential. And then with drug washout, the membrane potential will gradually start to recover to baseline. So our next question is whether MCH is acting directly on the cell that are recorded, or is it acting on something upstream from the cell I recorded? And so to address that question, we pre-treat the slice with tetrodotoxin. So tetrodotoxin will block any activity-dependent events. And so it takes away all the upstream events, and you're looking at just at the action of the cell you recorded from. So you pre-treat the slice with tetrodotoxin, and then in the presence of the tetrodotoxin, you also co-apply the drug that you're interested, or I'm interested in this case, MCH. And I can see that tetrodotoxin, in the presence of tetrodotoxin, MCH continued to have a membrane hyperpolarization, which tells us that MCH is acting directly on the cell that I'm recording from. And I know that this membrane hyperpolarization is in fact due to the activation of the MCH receptor, because if I now pre-treat the slice with an MCH receptor antagonist, that's called this TC-MCH7C compound, I just call it TC, we no longer see this membrane hyperpolarization. So the receptor antagonist, MCH receptor antagonist, completely block the actions of MCH. So we know that this hyperpolarization is due to the activation of the MCH receptor. Okay, so you understand now that MCH has inhibited the lateral septum cell. So how did this experiment actually work? So I'm just going to do a schematic to illustrate the process of how this works. So here's my brain slice. I'm recording from the lateral septum, and I've just expanded a couple of cells to illustrate this technique. Or the traditional technique. So here are my cells, and I approach a cell of interest, and I do a light suction to make a whole cell recording. And then when I have my continuous recording happening and I'm ready to apply my drug, I bathe the entire slice in the drug solution, and then I gradually wash out the drug solution. And so when the slice was bathed, that was what you see in the red, and then the washout continues. So I think you can see that when I apply the drug, all of the cells in that slice is contacted by the drug, right? The drug has basically contaminated the entire slice. So if I'm finished with this recording and I wanted to record from another cell because I'm having a really good day and I've got still some time left before I need to go home, if I wanted to do another recording, I have to change the slice and get another cell from another slice, and then repeat the process. Apply the drug, wash the drug out, so on and so forth, right? So this traditional patch clamp technique, the great advantage of it is that we're doing it in an intact brain slice. And that's really important because we keep all of those connections intact. And remember that the fibers have a very unique projection pattern, right? So in the intact brain slice, it's really important to keep together how is that drug going to respond to that neuron with all the other connections when it's intact. So I think that's an important aspect of brain slice recording compared to, say, cultured cells or dissociated neurons. The caveat of this traditional patch count recording, and also I should add that, patch count recordings are my favorite, but also the only way that we can assess the functional activity of a neuron. Is that cell alive? Is it excited by a drug? Is the drug causing the cell to swell or die very quickly? Or how chaotic is that drug effect on the cell, right? Electrophysiology allows us to see that basically immediately. The caveat of that is it's really time consuming. In the example that I showed you, it took about 60 minutes to apply MCH and see it wash out. Now, if I want to do the same, use the same cell and assess any pharmacology experiments, for example, if I want to apply a MCH receptor antagonist, or if I want to see if the receptor is activating a potassium channel or a chloride channel or any other drug application, it is a very, very long experiment. And, given that you're poking the cell with a sharp electrode and, basically creating a little hole, it's very difficult for the cells to stay alive, right? So if this experiment is going to take a very long time, you run a risk of not being able to complete the experiment you started. And because we have to change out the slice every time we want to do a new recording, then obviously this means that we're going to need more and more animals, right? The lateral septum, I think we get 2 really good lateral septum slices for mouse. So that gives me 2 recordings. If I want to do a third recording, I'm going to need another mouse. So you can see how it really ramps up our animal use. And inconsistency between slices, well, I showed you that the distribution of fibers and receptors will change depending on whether we're in front of the lateral, in the rostral part of the lateral septum, or if we're in the caudal part of the lateral septum. So if we're switching slices in between recordings, then we're getting a slightly different cell, right? We're getting a slightly different network. So how can Patch and Puff improve on this? So with Patch and Puff, the same setup as you do for the traditional technique, the traditional patch clamp techniques remain. So here you approach the cell, you perform your whole cell recording, and then instead of bathing the entire slice in the drug, you bring a puff pipette that you preload with a drug, and then you apply a gentle positive pressure like that to the cell that you're interested in, well, the cell that you're recording, and then you continue to see if that drug application has affected its membrane potential, right? So because this drug application is so short, it's very brief, it's only 1 second, it gets washed away very quickly. So the whole experiment can be completed in maybe 30 seconds, right? And now the next advantage is that if I'm finished with this cell recording, I remove my pipette, I bring in a new pipette, record from another cell in the same slice, and I just have to move my puff pipette next to the cell that I'm recording from. Okay, now I'm going to start my recording, and then when I do a pop application, I should see a change in its membrane potential, and then the drug washes out. So it would look something like this. Record the cell, I puff the drug, the cell responds to my drug, and drug washes out, cell returns to its baseline firing levels. So what does this look like in practice? In practice, here's an image of what that slice might look like that I'm recording from. On the right side is my patch pipette. This is the cell that I'm recording. And this is the puff pipette that I've placed, not immediately touching the cell, but a good distance. So 50 microns distance is what I typically aim for, to the left of that cell. And I also have prepared a video for what this puff system looks like. And to really illustrate the puff entering the tissue, I decided to preload the pipette with a fluorescent, like an Alexa Fluor. In this case, I think it's Alexa Fluor 647 or 540 something. So it's like a red color. In black and white, it's just white. So I've preloaded a fluorescent marker in my puff pipette just so I could see where the drug is coming and how long this lasts. And on the right side, it's hard to see on the dark, so I've outlined the dotted. I just did an outline of where my patch pipette is, and the cell is right here, but I confess it's really difficult to see that. So this is my imaging window, and below is my recording window. And this blue line is actually the live trace of my cell recording, and I'm recording the membrane potential. What you're going to see, so I'm going to just zoom in a little bit. Oops, sorry. Move forward. I'm going to just zoom in on the video so it's a little bit easier to see. When I play the video, you'll see the membrane recording continue, and then I would indicate when the puff starts. Now you're going to see a bright spot when the drug that has the fluorescent marker mixed in comes out. So it'll be really bright. And then you'll see that a few seconds after the puff comes on, there will be a membrane hyperpolarization that gradually returns to baseline as the fluorescence decreases or as the puff washes away. So I'm going to play that for you now. So here's the membrane potential recording. The puff goes on. A few seconds later, it starts to hyperpolarize. And then as the puff drug washes out, the membrane hyperpolarization returns to its baseline levels. Now, because I put the fluorescent marker in my puff pipette, the fluorescence is super exaggerated. But I hope you could see that the puff application was really just like a 1 second puff. The drug lingers for a few seconds and then will gradually gets washed out by the constant perfusion of the bath ACSF. Okay. Also, I should add, feel free to interrupt me if you have a question immediately. I do not see you, so just feel free to unmute yourself. Okay, so I've just shown you the video of how this patch and puff kind of works. And here I just kind of did this. So this is my Alexa Fluor that I puffed into the slice. You can see that it's touched the surrounding region, and I've recorded this. I filled the biosite into the cell I recorded from, just so I can demonstrate that I am recording the cell, and I am in fact getting drug touching my cell. Okay, so what does that recording look like in my experiments? So this is my sample trace of the membrane potential that received a puff in red here of MCH. And here's the quantification of the change in the membrane potential after receiving a puff of MCH. So we can see within a few seconds of starting the puff, we get this sharp drop in membrane potential that gradually washes out or returns to baseline as the puff of MCH is being washed out. I can puff this cell a second time, and this is what that looks like. So in this dark gray is the second time that I puff MCH, and when I quantify it, I do not see a significant difference in the change in the membrane potential, which means I can apply a second drug without much desensitization. All to say, I can apply this multiple times and still see a very robust agonist effect. So in case you're wondering, well, you've just done a big puff of a drug onto the cell, how do I know that the hyperpolarization is in fact due to my drug and not the physical pressure of more liquid or some kind of a current touching the cell? So I do a control where instead of puffing my drug, I just puff the regular ACSF that the slice is bathed in using the same process. And when that the puff pipette contains just regular ACSF, I don't see any change in the membrane potential. So by doing that control, I know that this response of my cell is specific to MCH, the drug, and not any mechanical movement of the application itself. And here's another example of doing pharmacology. Bring back in the TC compound again, which is the MCH receptor antagonist. So I show that my cell is responding to MCH, and then I can pre-treat the slice with the antagonist. And then when I puff MCH in the presence of the antagonist, nothing happens. So that's confirming that the cell I recorded from is in fact responding to MCH receptor activation. So remember I said that 17% of the cells in the ventral region of the lateral septum contains the MCH receptor. Imagine if I have to patch imagine, so 17% is, about 2 cells out of 10, right? I have to patch 10 cells in order to get 2 responders. If I do this using the traditional patch clamp techniques, that is a lot of slices and a lot of animals. It's going to take me a lot of time. And in fact, that is how we started. So I know for sure it's taking a lot of time because Michaela spent over a year trying to do this using the traditional method, but it was taking a very long time. So at that point, we transitioned into this patch and puff approach so that we can, within the same slice, patch a few cells before we need to change out the slice, right? So that allows us to have consistency within the slice that we're using. It definitely will require less animals. And because the application is so short, we can have multiple applications, and then each experiment can be also shortened. Now, for academic research, that's good, but I think even more beneficial would be if you were interested in pharmaceutical research or you're in pharma and things are moving very, very quickly. I think this type of work would be very beneficial. The caveat to this is, well, it's not a perfect system, right? When I puff the drug, it is not only touching the cell that I'm interested in. Of course, there is going to be a circumference of drug spread, but still, I argue that that small circumference is better than covering the entire slice with the drug, because then that would allow me to move my pipette to some other cell that's outside of that circumference and still do multiple recordings from the same slice. The other potential consideration is that even though I showed that in my hands, the puff application did not disrupt the patch, it is certainly possible that applying the puff could disrupt the patch. So sometimes when you apply the puff, you can maybe blow the cell away, right? And because the time course is so short, we can't really use the technique to assess the kinetics of the receptor. So some, I think, experimental applications for this technique are the following. I think it would be really beneficial for drug testing at multiple cell types. So imagine you've used some transgenic tools to label different cells within your brain. And in this case, I just made it the blue cell, the green cell, or the orange cell. You can move your pipette recording to different cells that are right next to each other and directly compare the response of that drug on 2, 3 more different cell types. So you've got this great internal control in your experiment. Pharmacology experiments are also possible, so I think there are not a lot of limitations in that regard. And thirdly, because the drug application or the puff application is so short, the receptor desensitization can be minimized. And then this process allows for higher throughput screening. So as I promised, I hope I've convinced you that the patch and puff application is a beneficial introduction to the traditional patch clamp technique. And so I'm going to talk about how we can go about doing this and showing you a step-by-step protocol. So the protocol for patch and puff is really divided into, well, I divided it into three parts. Preparing the brain slices, preparing the slice recording, and then this new part, which is the puff application. So if you already know how to prepare slices, or if you already know how to do slice recordings, this is all about the same. And then this is the only new part for you. But I think we all have slightly different backgrounds, so I'm just going to walk through each of the steps. So starting with brain slice preparation, when you first start preparing your brain slices, there are several solutions that you need to make. One is a ice cold cutting solution that we submerge the brain and then cut slices from. And then the second solution is a room temperature bath ACSF that we will recover the slices in. The main differences between these solutions is the amount of sodium, calcium, and magnesium. So in the cutting solution, this is a pretty stressful thing for the brain to experience. Excuse me. So you'll have low sodium, low calcium, high magnesium, just to prevent excitotoxicity. And it's really important to keep the brain as cold as possible during this cutting process because you really want to protect the brain as it's going through the slice. We use a microtome to cut these slices, and the brain slices might vary between 250 microns to 400 microns, but for patch count recordings, most slices are between 250 and 300 micrometers thick. So after you prepare your slices, you recover the slices for about an hour, and then you're ready to begin your slice recording. So when you're ready, oops, sorry, you oh, I lost that picture, but hopefully you saw that there was a rig picture that came up. You put that brain slice underneath the microscope, and then this is an example of what you would see under that microscope. So one really important step to brain slice recording is knowing how to identify a healthy cell. So I'm going to point out with yellow arrows some examples of healthy cells, which I hope you can see on the screen. So here's one, here are some, and what you want to look for in a healthy cell is that it should have a distinct shape, which means it might be oval or teardrop shape or be triangular, and they should be quite plump. So plump meaning like a pulp, so you can kind of poke it and be squishy. The white arrows I'm going to bring out are going to point to unhealthy or dead cells. So these unhealthy cells will appear quite contrasted from the background. They're typically dead, or you might have cells where you can see like a dark nucleus. I typically avoid those. So after you've selected a cell that you like, one of these plump ones, then you would approach that chosen cell with the patch pipette. And then your patch pipette is about 6 to 8 mega ohms resistance, so it's quite skinny. And it's back filled with another solution, the pipette solution. And that pipette solution is typically contains potassium, a high potassium solution. You then use a micromanipulator to lower your pipette next to the cell you're interested in. And one tip I have is you want to apply some positive pressure to the tip of your pipette, and that just really helps keep your pipette clean, so that as you approach the cell, you're always pushing away the debris and you have really good contact between your pipette and the cell that you're recording from. So up to this point, the procedures for patch cut recordings are fairly standard. So what I'm introducing in the puff application is really just one small addition. What you'll see is you'll have an additional micromanipulator that's carrying the puff pipette. So here is the electrode that's holding your recording electrode that you're doing the patch cut recording from. And on this side, you're going to place your puff pipette. And the puff pipette is you fill it with the drug that you want, and the puff pipette is actually just connected by a piece of tubing. A skinny piece of tubing that you have in the lab. And then this syringe is also a generic syringe, a 10 ml syringe that you might also already have in the lab. And To apply the puff, you can connect it to something fancier, like a mechanical puff, but we could actually do it just with a manual puff, using our hands. So it's a fairly cheap, economical way of adding this procedure too. So again, this is a picture of what that looks like. Your patch pipette recording from this cell. You place your puff pipette 50 micrometers approximately away from the cell that you're recording from, and then you apply manually, or I apply manually a 1 second puff, and then I record my cell. So in a nutshell, this is how we do patch and puff. And to wrap up my presentation, I'm just going to summarize what we discussed today. And first, that there is really significant interest in target discovery for obesity research to look for medications or new weight loss strategies. And secondly, I talked about how there are unique MCH projections within the lateral septum and then I found that within these unique projections from MCH neurons, those cells within the lateral septum are also responding to MCH. And using patch and puff, I was able to more rapidly screen for MCH responses. And I believe that this is advantageous because it allows for faster and higher throughput screening of drug sensitivity in the brain slice. So with that, I would like to thank our funders and especially all of you for staying with me and for your attention. I would be more than happy to take any questions you might have.